In the last lesson, we talked about how many neurotransmitters and drugs, like caffeine and nicotine, are amines. Nitrogen-containing molecules that are extracted from plants are called alkaloids. Caffeine and nicotine are both examples of alkaloids, but so are all the opioids. The opioids all act similarly biologically because they all have similar shape and functional groups. To try to treat opioid addiction, we need a molecule that has a similar structure and a similar functional group so that it can bind to some of the same signaling proteins in our bodies. But it needs to be different enough to not fully activate them. The molecule on the right is called methadone, which is the gold standard anti-withdrawal medicine for opioid addiction. It binds to some of the same locations as opioids. Notice that it has the aromatic ring and another aromatic ring, whereas the opioids have an aromatic ring and then two other rings. But it doesn't trigger the same high because the molecule is structured slightly differently. Our final functional group is the amide. Carboxylic acid, OHs, can be replaced with an amine, which creates an amide. Just like with amines, amides don't get separate functional group names for primary, secondary, or tertiary. They're all similarly reactive and are given the same name, amide. Carboxylic acids react with ammonia, which you can see in the first row, or primary amines, which you can see in the second row, or secondary amines, which you can see in the third row, in order to form amides in a very similar manner to how we reacted alcohols with carboxylic acids in order to make esters. In each case, the nitrogen and any carbons that are attached to that nitrogen will replace the OH on the carboxylic acid to make the amide. So the carboxylic acid still has the carbonyl and all the carbons attached in the same way, but instead of an OH, it has the nitrogen and its carbons. These reactions can be made to occur with really high temperatures, like we show here, 100 degrees Celsius, or acid catalysts, or enzymes. In the lab, we use temperature or acid catalysts. In our bodies, amidifications are performed using enzymes as catalysts. The most common amidifications are the formations of proteins from amino acids, which have both amines and carboxylic acids that can together make amides. We'll talk more about this process in our bodies in Chapter 22. While this is a common reaction in our bodies to make amides, our body can make very special conditions to get the reaction to happen, like a ribosome. If we just stick a carboxylic acid and an amine in room temperature water, we won't actually see that change. We won't form the amide. That's because even though the amide is relatively stable, the acid-base reaction between a carboxylic acid, which is acidic, and an amine, which is basic, is much faster and prevents the amidification from happening under normal conditions. This is good news in our bodies because we have a lot of carboxylic acids and amines floating around, and we don't want them randomly reacting. There are infinite conditions where we'd see the acid-base reaction instead of amidification. It's common practice to indicate room temperature for these reactions, either with the letters RT for room temperature or as 25 degrees Celsius like we have shown here, which is about the same as 77 Fahrenheit. In these cases, notice that every one of these carboxylic acids becomes the carboxylate, and every one of the amines becomes the ammonium. Look closely at the reactions that are shown in this practice page and try to identify the products. Remember that the conditions are important. A carboxylic acid and an amine are present in each of these reactions, but the temperature is different. The first reaction is an amidification. The nitrogen of the amine goes where the OH was, and it forms an amide. The second reaction is also an amidification, where the nitrogen of the amine replaces the OH. The final reaction is lower temperature, which means the amide can't form, and we have the carboxylic acid becoming the carboxylate and the amine forming the ammonium instead. Amides are hydrolyzed, that means water is added to break them into pieces, in the same way that esters are, but require heat, catalyst, or some sort of enzyme to do this due to the increased stability of the amide bond. When amides are hydrolyzed, they break up into the carboxylic acid and amine pieces that form them in the first place. That means that the carbonyl carbon and the nitrogen will be separated, and that the carbons all the way up to the carbonyl will become a carboxylic acid, and all the carbons other than the carbonyl carbon that are connected to the nitrogen will become an amine. So if we look at the first reaction, the carbonyl carbon is just one carbon off of the aromatic. In the product, those same carbons show up, but the carbonyl carbon is now part of a carboxylic acid. The nitrogen and the other carbons are a separate amine. See if you can identify the carboxylic acid and amine products of the last two reactions. There are two carbons up to and including the carbonyl in the second reaction, and these same two carbons become the carboxylic acid. On the other side, there's only one carbon connected to the nitrogen, and it stays connected to that nitrogen, becoming the amine. In the final reaction, the three carbons on the carbonyl side become the three-carbon carboxylic acid. The methyl and ethyl chain on the other side of the nitrogen remain on the nitrogen when it separates as an amine. In chapter 16, we talked about how lactones were cyclic esters. We can also have cyclic amides, which we call lactams. The most important class of lactams are the lactams which have the nitrogen attached to the second carbon away from the carbonyl, a beta-lactam for short. Beta-lactams show up in all the penicillin-related antibiotics. You should be able to recognize a beta-lactam as is shown on the bottom. When we name amides, amides are named very similarly to carboxylic acids from which they are derived. The carbonyl carbon will be the number one carbon, just like in carboxylic acids, and that means that the carbonyl side needs to be the main chain. The side chains on the nitrogen are numbered with N, just like in amines and in ammoniums. The amide names can also be thought of in relationship to the carboxylic acids from which they're derived, which can be helpful when we have systematic names for carboxylic acids that are a little bit different. Any carboxylic acid, common or IUPAC, will end in either ic acid or oic acid. If we want to make the amide, we just replace the ic acid or oic acid with amide. For example, if we have formic acid, it just becomes formamide. For example, the molecule on the left is derived from the carboxylic acid, benzoic acid. This name becomes benzamide. Again, this transition, changing oic acid or ic acid to amide, is useful for molecules with common names or systematic names that don't follow the rules, like benzoic acid. For other molecules, we'll just follow our normal pattern of naming molecules. The center molecule has two carbons on the carbonyl side of the molecule, making it an ethanamide. The methyl on the nitrogen makes it an N-methyl ethanamide. Try to name the final amide yourself. The carbonyl side of the nitrogen is a chain of three carbons, so propanamide. There's an ethyl and a methyl, making this an N-ethyl and methyl propanamide. There are two amide names that aren't systematic that I'd like you to know. The molecule on the left is called barbituric acid, but its conjugate base would be known as barbiturate. The entire drug class of barbiturates, which are used for anesthesia and to treat seizures, are based on this molecule. The molecule on the right is urea, a diamide that is the derivative of carbonic acid that allows us to remove excess nitrogen from our urine. And it's at this point, unceremoniously talking about urine, that we reach the end of our organic chemistry portion of this class. Congratulations, you did it!